This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on September 21st, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Today, we are recording in studio. We're in New York City. It is Saturday. It's a beautiful Saturday. Incredibly gorgeous day. It's no clouds in the sky whatsoever. It's 28 Celsius. Just a gorgeous day. And my guest happens to be in town for a meeting, so I grabbed him to sit down and chat about viruses. He's from the Laboratory of Structural Virology at the Institut Pasteur, Félix Rey. Welcome.
Well, thank you.
You say Félix or Félix?
Félix. My name is Spanish, so it's Félix.
Félix, with a little accent. But if it were French, it would be Félix, right?
It's Félix.
Félix, they pronounce the X.
Félix, the X is pronounced.
Okay. Welcome. I'm sure you've been to New York many times.
Yes. Yeah, of course.
So Félix was coming for a meeting, as I said. So I said, come by, and it's a Saturday. We'll record a podcast about his work. I want to start out by finding a little bit about your history and your training. I know you're from Argentina originally,
yes.
Right? You were born there.
Yes, born in Misiones, north of Argentina, near the Iguazu Falls. Mm-hmm. You probably heard about it.
Yeah, I've never been to Argentina.
Yeah, it's a beautiful country.
I'm sure. But I know a number of Argentine scientists, besides yourself, yeah. Raul Andino.
Yeah.
My old postdoc, Gerardo Kaplan, and I always meet them wherever I go.
Yeah.
So they train scientists well, it seems.
Yeah, there's a big diaspora of Argentinians. The problem is we get trained well, but then it's difficult to do science when you're in Argentina.
So when did you go to college in Argentina?
Yeah, I went to college. I studied theoretical physics in Patagonia. Actually, there's an institute called Instituto Balseiro in Bariloche. It's a beautiful place, a skiing resort. And then when I graduated, I wanted to turn into biology, and I ended up going to Paris and enrolled in university there.
It's for a PhD?
For a PhD, I did a PhD in structural biology. Well, knowing physics was kind of the easiest way to enter biology.
So how did you get interested in physics to begin with?
I ended up in Posadas, which is the capital of Misiones. There was the university. We had a college on chemical engineering. So I started that, and we had courses on physics. And then I ran into this book by Richard Feynman, the Feynman Lectures on Physics. And I liked it. And he was really also interested in biology, and he was even explaining the experiments of Louis Pasteur and the tartrate and the hand of these crystals. And so it made me like physics. So I applied to go to this institute in Bariloche, and I got a fellowship to go there. But then I realized that it seemed at the time that the most interesting things in physics were behind us. I mean, the first half of the 20th century. And in biology, everything was blooming. And so I wanted to learn biology after that.
What year was this, roughly?
Oh, I graduated there in December 1981. So, yeah.
You follow in a long tradition of physicists who moved into virology in particular.
Yeah.
Well, right? Max Delbruck, Leo Szilard, and others.
So, yeah, we're fascinated with this group theory, the icosahedral symmetry. So, things that seem to be so abstract in physics, then they turn out to be used by the real pathogens.
So you went to Paris to do a PhD, in structural biology.
Yes.
With who was that with?
I worked with Joel Janin. He was not really. He was studying protein interfaces, and he had a number of papers with Cyrus Chotian, understanding how proteins fold and how they interact with each other. But they gave me a project. I mean, I didn't have a fellowship at the beginning, but there was a company interested in producing high fructose containing syrup. And for that, they were using this enzyme, which is a glucose isomerase. It would isomerize glucose into fructose, which is an expensive sugar.
Yeah.
And so they say, well, why don't you, they were using it in an industrial environment at high temperatures to make it more efficient. And can you make this more stable? So I solved that structure, and then there was some that gave us idea of what to change, and they had a much more stable enzyme. But that's how I learned crystallography, and then at the time I was like there. This was in Orsay, and one of the people who had joined there because there is a synchrotron was Gerard Bricogne. And Gerard had been working with Aaron Klug and with Steve Harrison to use the symmetry, the icosahedral symmetry in the case of TBSV, or tubular symmetry in the case of TMV, to write up code so that they could use that to average the electron density and solve the first structures of viruses back in 1978. So I learned a lot working with Gerard and got the code how to do things. And since he knew Steve, he told Steve, maybe you should take this guy. And
Steve Harrison.
I went to Steve Harrison.
As a postdoc.
I was there for 7 years, yeah.
Okay. So, Gerard Bricogne was here for a while.
Yeah, he was in Colombia, I think.
Yeah.
Yeah,
that's where we are.
Yeah. Well, actually, he left Colombia to come to Orsay, where at the time when I was there, and so he's now in Cambridge.
In the UK.
In the UK, yes.
Yeah, he was here for a while, just a few years, I think. Yeah, and then he left. I don't remember where he went afterwards.
He told me his wife was not very pleased with the area where. At the time, maybe this some townships in New York where I don't know, things had changed probably.
Maybe. Yeah, he wasn't here long. I remember though.
Yeah.
So you went to do a postdoc with Steve Harrison.
Yes.
And I think that's where you worked on tick-borne encephalitis structure, right?
Exactly.
And then you were there for how many years?
7
years.
88 to 95.
Wow. You did some other things with him as well, right?
Yeah.
You did NF-kappa B, it looks like, right? I'm looking at your publication list.
Yeah. Yeah, we solved the NF-kappa B the same year, actually.
And then after that, you went back to Paris?
Yes. So I got a junior PI position at CNRS, which is the National Center for Scientific Research.
Right.
And so I started my own group. So and I was lucky. I met Gerd Wengler, who was working on Semliki forest virus, and he had isolated this E1, E2 complex, and which was, I got curious. Now, flaviviruses had this odd protein that didn't look at all like the influenza hemagglutinin. Let's try these other ones. And the big surprise was when we solved that structure, that it was folded just like the flavivirus, like the TBE. And so we introduced this class 2 of envelope viruses.
Right. So and then you've been in this same position ever since, right?
Well, no, I was at CNRS. This is Gif-sur-Yvette, which is a town 20 miles south of Paris.
Okay.
There is a big genetic center, and then I went to Pasteur. In 2004, they were looking for someone to take the virology department, and so I came as head of virology.
Is that the job previously held by Marc Girard?
Yeah, Marc Girard had been head of that department, but before me was Gerard Orth, who was working on papilloma viruses.
Okay. So I knew Marc Girard because he worked on poliovirus.
Yeah.
He was good friends with David Baltimore. In fact, last year we had an 80th birthday party for David Baltimore.
Yes.
And I saw Mark for the first time in many years. And he said he was his first postdoc. He was David's first postdoc.
Oh, I see.
When David went to, I think, the Salk.
Okay.
Early on in his career, and Mark was his first postdoc.
I see.
And he said, so they were working at MIT, and he said one day David came in and said, I'm going to Salk and you're coming. So Mark said, okay.
Okay.
You're coming. That was the beginning.
Wow.
Okay. And now you've been there ever since. And what I find remarkable looking through your papers, you've solved structures of different virus proteins from all different families, not just flaviviruses, alpha viruses, some reoviruses as well.
Yeah, rotavirus.
Rotaviruses. Yeah. You were the first to do the Hep C RNA polymerase.
Yeah.
I remember that very well because the first author, Racaniello,
yes,
the name kind of similar to mine, with a vowel at the end. Is he Italian?
No, he's French, but probably his family were Italian immigrants went to France.
But that was really an interesting polymerase.
But we were collaborating with the Italians for that structure with Raffaele De Francesco, who was the first one to characterize the polymerase.
And you've done VSV, Simliki forest virus, Berna viruses, Picoberna viruses, I think.
Yeah, we were curious with the
and more recently, Bunya viruses. But I thought we could focus on flaviviruses and talk about those a bit today. So let's go back to the tick-borne encephalitis. So why did you decide to do that structure? But at that time, the flu hemagglutinin was the only viral glycoprotein we had, right? Yeah. So why did you decide to do TBE, which is a flavivirus, right?
It's a flavivirus. It turns out, no, it's because at the time we didn't know how to make these recombinant proteins at the time, and you needed to pick up a system in which will be a way of producing enough of the protein so that we could make crystals of it. And we were, Franz Heinz in Vienna was very impressed by the work on influenza, and they were doing a vaccine for TBE, and so they were making a lot of that protein. And so Franz was the one who showed that you could solubilize them with detergent and treat with trypsin, or he was treating directly the particles with trypsin, and he could get a very stable fragment of the protein, most of the ectodomain. And so he had been sending them to Steve already. But when I got there had been some crystals, but they were not very good. And so I took on that project. It was my second project. The first one was TF3A, the transfixion factor 3A, which has 9 zinc fingers in complex with a 7S RNA. And that's something that still today has not been solved. So luckily I had something else to work on and results.
Was it hard to get the structure, get crystals and the structure? Well,
at the time, because it's not like today when you get crystals like the FRAC today, you have all the methodology is developed that it's straightforward to get a crystal. At the time, it was not the case, and these were very tricky crystals. They were twinned. It means that they were not single crystals, but a mixture of two, and it was very difficult to disentangle them. And so we ended up having, at the time, we didn't know yet the methods to cryo-cool the crystals to have them so that you can collect a complete data set from a single crystal. We were just taking one shot, another crystal, and again, and put all that together. So it was very time consuming. We're using films. Now you just have direct detectors. You record it. So,
I remember the films.
Yeah. So it was quite laborious. And so I struggled with that, but then in the end, it paid off.
Many people in the world know films because of the famous X-ray 53 from Rosalind Franklin, right?
Yeah, exactly.
The one that Watson and Crick used to say, this has got to be a double helix.
Yeah.
Films. I knew Jim Hogle early in my career, and
Well, he saw
using
poliovirus.
and he was using films at the time.
Yeah.
So did you have some reason to expect that the glycoprotein would be flat on the particle?
No, we had no idea. So when we saw this dimer, so we postulated it was the curvature. I would match the curvature of what we knew these flaviviruses should have. So this is, but it was only when Michael Rosman and Richard Kuhn came up with a cryo-EM that had enough resolution that this dimer could fit, that really validated.
How long after your structure was that?
Oh, that was 2002 probably they published that. So we had from 95 to 2002. So they fit the TWiV structure onto the dengue.
Right. And that was the first time you realized it was flat on the surface.
It was but we suspected.
You suspected it, yeah.
And so we had a model, but we would thought that we'll have a T equal 3 or something, which it doesn't. And it's a very, very interesting herringbone pattern of 3 parallel dimers. But you need to. So we had with Steve Fuller, we had worked on cryo-EM before that on virus-like particles. If you express just the envelope proteins PRM and E, you get particles secreted. But these particles were very heterogeneous. So Steve Fuller had only took the smallest ones, and he got a very smooth sphere there. So, and we placed 30 dimers to make 60 on a sort of pseudo T equal 1 particle. But you could fit it. It was supposed to leave holes that we didn't see on the reconstruction because it was just flat.
I see.
So,
and then you said when the Semliki glycoprotein was solved, it turned out to be the same. It was flat. Exactly.
So it's it's not as that it's flat. The Semliki is more spiky, but there is a heterodimer there. Is in the case of flaviviruses, the companion protein, all these class 2 proteins have a companion protein that play a little role as a chaperone. So they are in a polyprotein and they are translocated first into the ER lumen. And when the fusion protein is translocated, the other one is already there and grabs it while it's folding.
Yeah.
So then they are transported to the surface and this companion protein gets cleaved by furin. And so you release... in the case of alpha viruses, is a much bigger protein which is responsible for receptor binding and which stays bound. So instead of being flat, it's like at an angle, like 45 degrees.
Yeah. Right. Now, in these flavies, they have an RNA, a plus strand RNA, and there's a protein surrounding the RNA.
Yeah, completely coated.
And then there's a membrane.
Yes.
And then the glycoprotein.
Yeah.
Which the glycoprotein by itself makes a shell, basically.
Exactly. So this is a property of all of these class 2 proteins. For alpha viruses, it's the same. And for the Bunia viruses, we're finding that it is the same.
It makes a shell which is kind of flat.
Exactly.
Right?
Interesting. Exactly. Yeah. Interesting because there was a paper a few years ago on the S-layer protein of an archaea.
Yeah.
And that S-layer protein, the only homologous protein that they could find by search was these class 2 envelope proteins, the central domain, and that S-layer makes a layer and gives curvature to the surface of the archaea. So maybe there is some very ancient origin of these proteins.
So for the flavivirus E proteins, what is the T number of the symmetry? Is it T?
Well, it's not a triangle. It doesn't follow quasi-symmetry. So you have 3 dimers that are parallel to each other, but each dimer, its environment is totally non-equivalent to the others. Okay. So it's really not, it doesn't follow Casper and Klug.
But it does make a repeated structure.
Exactly. Right? You have 3 dimers. You have 30 faces with these 3 dimers, parallel dimers, diamond shaped. But there's no, the T numbers come from quasi-equivalence.
Right.
And here there's no quasi-equivalence. There are no 6 folds and 5 folds that you will see.
And this arrangement is entirely a consequence of the way the proteins interact. It has nothing, there's no contribution from the proteins underneath, right?
Yeah, exactly. So the budding is still not really understood because there is the capsid protein that binds the genome, and then the glycoproteins they don't have a cytosolic tail. They have a transmembrane hairpin, and it's just the turn of this hairpin that is cytosolic. But apparently they form a bundle of alpha helices that is somehow recognized by capsid dimers that bind to the RNA.
Oh, so they do interact with the capsid protein.
Yeah, exactly. But this is an immature particle that buds in the ER, which is a spiky particle. It's not flat at all. And this particle is transported then, I think someone showed that they interact with the KDL receptor to begin the journey across the exocytotic pathway. And then the acidic pH in the Golgi, in the ER, the pH is neutral. And but these particles, the immature particles are very sensitive to pH, just like the mature. But so when they reach the Golgi, there is the major conformational change. The spikes that are trimeric spikes dissociate, and the envelope proteins make these dimers, and the companion protein pre-RM exposes this site specific for furin, and furin cleaves there. And so when they are the part that is cleaved from PRM remains bound as long as the pH is acidic. But when the particle is released, the pH is neutral again, and the affinity is weakened, so it's shed. And then the next time this flavivirus particle will encounter acidic pH will be in the endosome of another cell, and there the fusion
occurs. Right. So the capsid protein itself, that has no symmetry at all. Right.
Well, no, all of the cryo-EM structures that have been published, they have not seen, except for an immature Zika virus structure in which they could see some density underneath this bundle of TMs of. Okay. But it was an eccentric structure, so it was reported very recently.
But is that the same in the alpha viruses, or is there some?
No, the alpha viruses, they have a very organized core with T = 4, right. Which matches the symmetry of the glycoproteins on the outside.
So in those viruses, the glycoproteins assume icosahedral symmetry because of the underlying capsid.
Well, no, a mutant was shown. There was a mutant of the capsid protein that couldn't make this this internal nucleocapsid by itself. But if by binding to the tails of the glycoproteins, you could rescue virus. And when they did the cryo-EM, there was order outside by node. This was done by Henry Garoff and colleagues some time ago. So apparently, it's the glycoproteins. There is a tail that will interact. There are 240 of these tails, and there are 240 binding sites on the nucleocapsid that is formed by the capsid protein. Those interactions are what drive. So if you break the lateral contacts between the capsid proteins, the binding to the tails will rescue that, and you get more fragile particles, but they're still viable.
So the glycoproteins can still organize properly.
The wild sports, yeah. Right? I think this is the characteristic of all class 2 viruses, is that they drive the budding. So there's no escort that has been shown to. So and the fact that if you only express the glycoproteins, you get budding of closed particles is sort of telling you, although they are irregular because the genome is probably making a size, a radius that is more unique.
So class 2, you can produce the protein and will make a particle. But you can do that with HA also, no?
Well, yeah, but it doesn't drive the budding. I think the HA...
You still need escorts,
yeah.
Okay, got it. So what are the other properties of class 2? They're flat on the membrane, right? Or parallel to the membrane, as opposed to class 1, which are perpendicular.
Yeah.
And what else do we know about class?
Well, the class 2, this capacity of driving budding or making a closed particle by lateral interactions, and they are so not all of the class 2 viruses we know today are icosahedral. They all make a scaffold outside, but for instance, hantaviruses do not make it. They make pleomorphic particles with square spikes, or a tetramer instead of a trimer, like in alpha viruses.
Okay.
They are. We don't understand how they drive budding, because again, you just express those glycoproteins and you get, and there is no matrix protein there that is underneath. So it's okay. But what is interesting on class 2 is that we are finding them in cellular proteins.
Right. In fact, these are gamete fusion proteins.
Gamete fusion, yeah.
You solved one of these structures.
Exactly. So, well, the first structure we solved was not a gamete fusion, was a protein of in C. elegans that is responsible for making the skin formation. So I remember I was given a seminar at NIH. I talk about the post-fusion form of the Semmelweis virus class 2. And at the end of the seminar, someone stepped to me and say, we identified the first bona fide cell-cell fusion protein, and it's predicted to be all beta sheets like yours. So I'm curious. So I got interested in that. This was Benny Podvilevich. And so 10 years later, in 2014, we were able to solve that structure and show that it was just like the others. And so there must be some common ancestor. And now the gamete fusion brings this much further out, because it's a protein that is present in 4 out of the 5 kingdoms of eukaryotes. And so many people think that it was present in the last eukaryotic common ancestor, the LECA.
LECA, right. It depends how you think viruses evolved. Do you think that was then acquired by viruses for fusion, or did the viruses have it before LECA?
That is the question. So somebody wrote a comment, who came first, the virus or the egg? So the thing is, what this shows is that the impact of genetic exchanges between viruses and cells, we're seeing it all the time. You know, the CRISPR is just that. And during the whole evolution of eukaryotes, but probably of cells, there have been exchanges with viruses.
Sure.
And you know, the life became, the primordial cells were some, at the times there were not lipids, there were some fatty acids that would be stabilized by some protein precursors of some amino acids or something. So proteins that interact with the membranes must have been at the very beginning, and they have been a virus cannot survive without a cell, and it's difficult to say now if the cellular protein is a viral protein.
But no class 1 fusion proteins are found in cells, right?
Yes, there are. Syncytins.
That's right. Those are given by retroviruses.
Exactly, right? But this is the same. That's retrotranscription is like the way to insert something into the genome of a cell. So, and the class 2 proteins that I told you in C. elegans is class 2 proteins. Harmeet Malik had shown that C. elegans retroviruses have class 2 proteins. And so actually his theory is that retroviruses evolved from retrotransposons that picked up an envelope gene to go from one cell to another. And this was done independently on different cases. So vertebrates had class 1, other organisms had class 2 and even class 3 on some of them, looking like the GB from herpes viruses.
But contemporary retroviruses have class 1?
The ones those that infect vertebrates, yes, have class 1. But for other organisms like cats, or there are many, they have different glycoproteins.
So the ones that infect vertebrates, presumably those glycoproteins also came from the vertebrate at some point, or maybe some other organism.
Yeah, some other.
But we don't know the origin.
We don't know where. So it goes to show. I mean, there are very few. I mean, all of the structural studies today have shown only 3 different classes of fusion proteins from viruses that are completely. For instance, class 3 are on herpes and on rhabdoviruses and on Borna viruses and on Togoto viruses. So there are it somehow the genome of a virus is like a mosaic of different proteins beginning, those proteins for replication come from one pool, and those for transit to other cells come from other pools. And it's also shown that probably a protein that can fuse membranes, it's something quite complicated. And instead of starting from scratch, nature uses it over and over again. So it's, once you find a solution, there is no reason why to.
Right. When you first saw the TBE glycoprotein, and as you said, you predicted it would be flat.
Yeah.
Did you think about how it would catalyze fusion at that point? What were your ideas?
Well, because it was a dimer and all of the fusion protein and trimer. So no, we immediately started to try to understand how it would catalyze fusion. So the two structures that were there were class 1, were influenza, and Ebola, I think, was one of the first ones that showed that they make a hairpin. So we ended up with Franz Heinz on tick-borne encephalitis virus, and with Margaret Killian working on the Semliki forest virus, because she was able to show that there was a trimer that only after you expose to acidic pH, the two glycoproteins E1 and E2 that you have on the alpha viruses separate. And so we worked on parallel, and Steve did the same with Steve Harrison with dengue. And so the structure showed that it makes a hairpin as well. So the fusion is not a fusion peptide, it's an internal fusion loop, and it's not at the end terminus like in class 2, but is at the distal end of one rod that is all beta sheet, but is a rod. And then the third domain and the region leading to the transmembrane come back. So they make a hairpin. So although they have a very different structure, they drive fusion probably by a very similar mechanism.
And it's low pH driven.
Is right? Low pH driven.
And so then you solved the low pH structure of the TBE glycoprotein years later.
Yeah.
Right.
It was also a Bressanelli paper.
and that showed that, in fact, that low pH, these changes occurred.
The object changed the conformation to make a hairpin, just like we had seen before with influenza hepagglutinin that changes to a hairpin.
Now, for the type 2, does the fusion loop have to be cleaved, like the flu has to be cleaved to make an N-terminus?
No, the class 2 are different. The cleavage is on the companion protein, the chaperone that gets cleaved. But that's true for alpha viruses and for flaviviruses. The Bunia viruses, there's no cleavage, and we don't understand you need to ensure that your protein will not be active prematurely.
Right.
And so this is how they are made, with this second protein that gets cleaved. But Bunia viruses and also rubella virus, which has a class 2, they don't have such cleavage. So we don't understand yet what is different from the virus as it's been made, or at least the fusion protein, and when it enters.
So for the Flavi E, the second protein hides the fusion protein, so it doesn't fuse with anything.
The second one, yes.
What's the name of is that PRM? Is that
PRM? Yes.
Okay. So that's on the surface of the particle.
The surface, that's an immature particles. Then it gets cleaved, and when it's cleaved, the fusion loop is hidden at the dimer interface. So the E proteins make a dimer with the fusion loop at the interface. Okay. And this fusion loop, this dimer is kind of not very tight dimer. So there is exposure of the fusion loop, and the fusion loop is very conserved. It's across flaviviruses. This is the most conserved region, and we suspect that this is one of the reasons that we have in dengue for this so-called ADE effect, antibody-dependent enhancement, is that if you are infected by one serotype of dengue virus, then it makes a lot of antibodies against the fusion loop, and those are the only ones that will interact with the second serotype later on. But if you have a particle that only exposes the fusion loop at a few spots out of the 90 dimers, or just a few, then you will bind antibodies, but not in enough stoichiometry to neutralize the particle, but they will be very efficiently incorporated into immune cells of the immune system via the Fc receptors, and infect much easy because the particle is not neutralized.
So basically, this is like a universal epitope, like we talk about influenza stalk epitopes are universally considered.
Yeah, except that it's different because here is an epitope that you don't want antibodies against it. So the vaccine, a safe vaccine should be a vaccine that doesn't elicit the antibodies against this epitope because because of these serotype things, it's being a problem more than instead of in influenza, it's something you want because this allows you to neutralize a broad amount of viruses.
Okay, so for dengue and other flavies maybe, but predominantly dengue, you do not want antibodies against this because they will cross-react and cause
They will cross-react without neutralizing the other particles. Just because the fusion loop is not exposed throughout the particle, it will be only exposed in some few dimers. So, this is complicated because lab-adapted strains, they expose the fusion loop very easily, but the field strains circulating, they are much more sturdy. So it's much more difficult to neutralize with these fusion loop antibodies to the circulating virus that and this was the Sanofi vaccine. So they had titers, neutralizing titers, but it depending on what strains they were using to. So it is a tricky problem. So we think that if we could generate an immunogen that does not expose the fusion loop, it will be a major advance in avoiding having this type of antibodies.
So the Sanofi vaccine, the dengue vaccine, it was a tetravalent, but the type 2 was the problem.
Yeah.
Even though it generated neutralizing, is it right? Neutralizing antibodies? It's
those were the ones more difficult to, and serotype 2 is for some, in all our experience seems to be the one that is less well structured.
So the loop is exposed more.
It's more exposed, it's the one that causes more problems. So I will not say that we understand exactly what is the thing, but we suspect that it has to do with this breathing behavior and exposure of the fusion loop.
So the problems with the type 2. So a number of kids in the Philippines, right, had been immunized, and then when they got infected naturally, they had very severe disease. Yeah. And is that because the type 2 antibodies are recognizing this fusion loop? Or
I suspect that if this vaccine was not generating antibodies against the fusion loop, you wouldn't have that problem. But that's a hypothesis. We need to prove it.
Because that vaccine generated fusion loop antibodies, then when they were infected, those caused problems because they broadly bind but don't neutralize. They will bind another serotype.
Will neutralize best the other serotypes, but not the serotypes.
So why does it not neutralize? Is it different enough? The sequence different enough between serotypes?
No, what I'm saying is they bind to the fusion loop,
right?
But the exposure of the fusion loop is uneven. So if you expose it sufficiently regularly around the particle, you know, there is a Ted Pearson show that you need a stoichiometry, you need at least 30 antibodies coating the particle to get neutralization. But if you cannot reach, even if you have a high number of antibodies, they will be able to bind only to the dimers that are exposing the fusion loop.
Yeah.
And those may not be enough. So you are putting antibodies on the particle in not enough amounts to have that particle neutralized. So they will be incorporated into the endosomes, but they will not be neutralized, so they will infect.
So particles are made that have different numbers of fusion loops exposed? Is that what you're saying?
No, the particles have there are 180 E proteins.
Right.
That means 90 dimers.
Right.
And each fusion loop in a very well-conformed particle is hidden at the dimer interface.
Okay.
The problem is that some of those dimers will expose that fusion loop, and then once the antibody grabbed it, the affinity can be very high and it doesn't release it. So you will have that one antibody bound there, but all the rest cannot bind because they are closed. Maybe with time you will be a second dimer that opens, and then but there is a certain time kinetics that these particles will have this tag, that is the antibody with the FC, that they can be incorporated and brought into the endosome before having the chance to be completely having enough antibodies such that it's neutralized.
I see. And if it's a different serotype infecting you, the memory is against the original infecting serotype. So those non-loop antibodies won't even bind well.
Won't bind.
But the loop antibodies will bind.
Will bind. Yeah.
They won't neutralize because there are not enough of them, or
because not enough of them can. So there can be enough, but there are not enough sites. So you see what I mean? The surface of the virus particle will expose only too few places.
Okay. Got it. But it's enough to grab with an FC receptor.
Exactly. You just need one, and yes.
Got it. So the ADE, antibody-dependent enhancement,
yes,
is explained by these fusion loop antibodies.
This is what I think explains it, but I don't. We were trying to prove this now, and this is the kind of things. It's an interesting approach because this is an example of understanding the structure. The structural biology can lead you to understand a problem like this, the severe.
And that's why you solve structures of antibodies bound to.
Exactly. So because, this is our collaborators in the UK, at the Imperial College, and now in Oxford, Gavin Screeton and Tutatip Monkosapaya, they had this cohort in Thailand, and they have isolated some antibodies that are extremely powerful and which are neutralize all 4 serotypes. So we were, if you could generate those, and we spoke that they would protect.
Where are those epitopes, do you know?
Those epitopes are at the dimer interface on top of the fusion loop, but with the residues of the fusion loop are hidden at the dimer interface, and they come uncoated there. So they extremely neutralize because the dimer cannot open. And that region, there is a patch there. It's exactly the same patch where PRM binds in the immature particle. So it's difficult to get escape to those antibodies, because then you need to change also PRM to be able to bind.
So those are different from the fusion loop antibodies.
Those are different. The fusion loop require dimer dissociation to expose the fusion loop fully.
Okay.
And these are...
So how can you induce antibodies to this conserved?
Well, we're working on that. We are engineering disulfide bonds to the dimer, and allowing this so that it doesn't open, and we are trying to drive. So, for instance, there are different strategies. If we want to focus on that, we could do some epitopes focused vaccine design, in which you can resurface other regions and expose on. So
I remember you talked about that in Rotterdam, putting the disulfides into to prevent exposure of the fusion loop.
Yes, exactly.
I see. And so then no matter what kind of an immune response you got, those antibodies would not be able to bind the fusion loop, so you would not get ADE.
Yeah, exactly. So we want an immunogen, if you are going to vaccinate people, that does not expose the fusion loop. And this was a way we are mutating the fusion loop and changing to a cysteine, such that at that contact with the fusion loop, there is another cysteine right in front, so it cannot open.
But if people are naturally infected, they're going to have antibodies against the fusion loop, right? So then when they get a second infection, you still can have ADE, right? So the vaccine won't prevent that.
maybe the vaccine, if it's not exposed in the fusion loop, it's triggering some additional antibodies which would be protecting.
Right, so the key is you have to immunize them before the second infection.
Before, exactly.
Right. Okay, which can be done. You get them young enough, right?
Yes.
Interesting. Now, does this happen with Zika? Is there ADE? This is a controversial area, right?
It's a controversial area, yeah. I don't think people have really shown it in vivo, but we don't know what will happen if you introduce now a vaccine against Zika virus, because it will probably protect the Zika, but we don't know what this will be doing to future dengue infections.
I know
that is an issue.
If you have in vitro, in cell culture, people have shown dengue antibodies will enhance Zika, right? Yeah,
exactly.
And have they also shown vice versa?
Yeah, I think but Zika is more, they have also shown that these fusion loop antibodies that you can manage to neutralize dengue, but you cannot neutralize Zika with those, because Zika is less of, is not as dynamic in the envelope as dengue is.
And of course, with Zika, you have one serotype.
Yeah, there
also,
are no,
so there's
exactly.
no Zika ADE.
No. But the problem could be for dengue if those antibodies that you raise that protect you from Zika help. So, but there again, the vaccine against Zika could also be engineered not to expose fusion loop, and you don't want anyway those fusion loops, and then it should not be a problem.
So I think in people, there's no evidence for Zika dengue AD.
No, I don't think anybody has really been able to show that.
Right.
But Zika is not, well, in a pregnancy model. So will antibodies against dengue enhance Zika? No, but we don't know the opposite either. So I think there's still some work to do there.
But for Zika, the structure is similar. There's an E-glycoprotein. It's a type 2. There's a fusion loop as well. Right?
Yeah.
And is this exposed on particles?
Well, this is because, it's the dynamic behavior of these dimers depends on their overall sequence, and they are not identical in sequence. And it turns out that the E dimer for Zika virus is much less dynamic than the one for dengue virus, especially dengue 2.
You published this paper in 2016, Structural Basis of Potent Zika Dengue Virus Antibody Cross-Neutralization.
Yeah.
What is that?
No, but we show that this particular epitope, these antibodies that were neutralizing all 4 serotypes of dengue also were potently neutralizing Zika, and even more potently than dengue. So
and that's that interface.
Is exactly that interface that I mentioned. But then we try if they would be able to neutralize other flavivirus, but they don't. They don't neutralize yellow fever.
So that's the dimer loop interface.
Exactly. The dimer at the fusion loop interface. Yeah.
Okay, so those are good. The good antibodies. Those are the ones you want to induce somehow.
Exactly.
So somehow a vaccine has to induce those.
Yeah.
I understand that there's an infectious attenuated vaccine going through trials now. Right, it's an NIH developed vaccine, but it's not manipulated in any way to affect the epitopes, right? So you could have the same issues, I think, right?
Probably. Although I don't think they will have problems protecting against Zika. So, but the issue could come with dengue.
You're an author, at least, on another paper, a protective Zika dimer-based subunit engineered to abrogate antibody-dependent enhancement of dengue infection. So is that where you put disulfides?
Yeah, exactly.
So you will not get antibodies against the fusion loop?
Against the fusion loop, yeah.
Okay,
so those
got it.
All the same story.
Now, you mentioned earlier the production of particles, and you have actually a review article called Flavivirus Structural Heterogeneity. So maybe tell us how these viruses, you mentioned it before, let's go through it again. How do the viruses, how are they produced in cells, what happens, and then in the end, how are there different particles? You talked about spiky particles and so forth. What's going on?
Well, so this, I told you that the flavivirus particles bud in the ER lumen,
right?
And they have a completely different organization with 60 spikes with the PRM capping the fusion loop, and those spikes they undergo this conformational change when they reach the Golgi because of the pH, the acidic pH of the Golgi, neutral pH in the ER, and then in the Golgi they undergo furin maturation, which will cleave PRM for the release. But what happens often is that the maturation by furin is not complete. So you get parts of especially furin is a membrane-bound protease. So the side of the virus particle that is against the membrane gets cleaved, gets mature, and the opposite side is not. And then it has been shown mosaic particles, and this was work done at Purdue by Michael Rossman, showing that these particles have, one side spiky and the other not, and exposing, and in the spiky side, the fusion loop is exposed. So those is one of the reasons why they are, because the fusion loop is very immunogenic and is highly exposed there. You get a lot of antibodies against that.
I see. So as they're coming from the ER, the E glycoproteins are raised. That's why you call them spiky. And then as they pass through the Golgi, low pH and furins,
they lie down.
They lie down.
They lie down on the Golgi. But this is the lie down is not all of the class 2 proteins. Is specific for the flaviviruses in their mature form.
Okay, and then you're saying it's not 100% efficient process.
Yeah.
So you get production of particles that are mixed, mosaic.
And also, we looked at the furin side. The sequence specific for furin is suboptimal. The virus maintains it in a suboptimal way because of a strategy of maintaining, having all of this heterogeneity appears to be selected for by dengue.
Do we understand why it's selected for?
Maybe because it allows it to enter more efficiently via this antibody dependent. So there is some interaction. So
interesting. So if you're making a vaccine, you don't want to have spiky particles.
Exactly. You want to have the particles as smooth as you can, and the more static. I mean, to kill this this breathing behavior.
How do you do that? Do you treat them with furin?
Well, you can overexpress furin, but also even that doesn't lead you to 100%. So some certain flaviviruses get much more easily. And it also depends on the cells. Some cells have high furin, other cells have little furin. So in your body, depending on where those particles were made, they will not. So the idea would be to have something that exposes a very regular the E protein only without any M. There are some antibodies against this PRM that it remains, and those antibodies also enhance. So there are two things: fusion loop antibodies and PRM antibodies, which are.
So your strategy of putting a disulfide will prevent this loop from being immunogenic, right, or being recognized.
Yeah, but then we will. The thing is, how do you make? We can make the recombinant dimer, but another thing is to have a particle exposing the E dimers the same way as on a mature particle too. And that is tricky.
Yeah.
Especially because of this funny assembly that has to go through immature particles that then gets cleaved on.
And this happens for both dengue and Zika, this production of spiky particles?
Yeah, all of flaviviruses have that, except that for Zika or for all the others, the cleavage site is much more accessible. Dengue has this site that is apparently, if there had been a selection for have a suboptimal, you need to have furin cleavage because it's required for infectivity, but if you maintain at suboptimal, it's enough for the virus, and apparently that's how.
Your strategy of putting disulfides in, that would be for a subunit vaccine, right? Protein
for a subunit.
And that would lock
because you will kill. You cannot have.
Can't make virus.
You cannot make virus because it needs to open to infect a cell.
Right, and then that epitope is hidden. So have you immunized with animals with that and shown that you don't get dimer interface antibodies?
Yeah, no, this is the last paper you mentioned.
And those now will not cause ADE because they cannot bind. There's no antibodies made to bind that fusion loop.
Exactly. Right? So this is our hypothesis, I'm telling you. So what we predict is that that's what we want to show. Yeah.
Okay. And then so you immunize people with that, and even if they've had already a dengue infection and now they have antibodies against fusion loop of one serotype, you now immunize with this glycoprotein, the dimer glycoprotein, and this will now make neutralizing antibodies against all four serotypes, and there will be no fusion loop antibodies made.
Yeah, no additional fusion loop antibodies. You will not be adding.
No additional ones.
What happens is often on a second infection, the cross-reactives are those, and so you will be making more of those.
And so if the person is challenged, they will be immediately neutralized by all the antibodies that are produced, and there are none to be directed against the fusion loop. That would be a problem. So is this going to go into people at some point?
Well, we're not there yet.
Yeah. That's the plan.
Yes.
Yeah. There certainly is a need because the Dengvaxia is not good, right?
Apparently, they're having I think they in countries like Brazil, they have slowed down the number of hospitalizations, except that overall. So the problem is for children I think they're not giving it to children under the age of 9.
That's right.
In a way, so that they know that it's
right. Yeah. So that way you've it's not going to be your second dengue infection, right?
Yeah.
The other issue with that vaccine is that it's a yellow fever virus with dengue glycoprotein, right?
Exactly. So that's another. So I know, apparently most of the T cell epitopes are against the non-structural proteins, which here are all yellow fever, for instance.
Right. So these kids will be protected against yellow fever, right?
Yeah, maybe it's a very good vaccine against yellow fever.
Yeah. Well, I think you also need glycoprotein antibodies for that, right? Because the yellow fever vaccine, but it's very interesting.
Actually, I'm not sure people know exactly what makes yellow fever vaccine such a good vaccine.
Yeah. Well, it's the whole virus attenuated, right?
Yeah.
So you've got glycoproteins plus non-structural T cell epitopes. So,
and there's also NS1.
Yes. And
NS1 is the marker of infected cells because it's the only protein that goes to the surface. This non-structural protein 1. Mm-hmm. And so ADS that could play a role also by killing infected cells, which by budding into the ER, the envelope proteins are never exposed at the surface of the infected cell.
That's unusual, right? Because typically glycoproteins influenza glycoproteins are on the plasma membrane. Yeah. Well, that's
exactly.
a good strategy for evading T cell responses.
Yeah.
Right?
Yeah.
So NS1 is on the surface, you said? It's also secreted, NS1, right?
It is also secreted, so it's not an integral transmembrane protein, but there are dimers. It's secreted as a hexamer, and it carries lipids, like a number of lipids. We suspect that it has a lot to do with the pathogenesis, the NS1, and it's like a lipoprotein.
And you've solved structures of NS1s, right?
Yeah, we did a cryo-EM structure of NS1 showing that it's carrying lipids and 3 dimers. So the membrane-bound form is a dimer. And then 3 of these dimers hide their hydrophobic part, but they pick up lipids with that.
Right.
And they're transferred.
So in the years that you've been doing crystallography, have you witnessed this shift from crystallography to cryo-EM, right?
Yeah. So I consider myself a structural biologist. I don't mind if it's not a crystal. I mean, actually, it's so much easier. I mean, sometimes we struggle. We spend 10 years to get crystals that diffract well enough of these glycoproteins in order to get that structure. So now if you can get to the same resolution by a method that doesn't require that,
but is there a lower limit to the cryo-EM?
Well, yeah, but I think they are going lower and lower these days. So things are evolving very fast.
So do you still do crystallography at all?
Oh, yeah, we do because we are using cryo-EM for the things that are more tricky, that there's no way we're going to get crystals because and also what is very useful now is doing tomography, because so far by cryo-EM we could get to high resolution of particles that are all identical so that you can average them. So they are cosahedral more. But these pleomorphic particles like hantavirus, so there are people like Juha Huiskonen who are working on hantaviruses and getting these pleomorphic particles to pretty high resolution. And you probably follow John Brakes with the HIV core particles. He was able to reach close to atomic resolution by doing subtomogram averaging, and the field is evolving very quickly.
And as you probably know, here Joachim Frank is right across the street.
Yeah, sure.
We got last year the Nobel Prize for helping to develop these methods.
With Richard Henderson and with what's his name, the first cryo pictures at EMBL, the third Nobel Prize.
Well, let's look it up. Nobel Prize from last year. I was in Stockholm a couple of weeks ago, and I saw their pictures on the wall there. I know Francis Arnold was one of the other ones.
Dubochet is the one I was looking for.
Is that right?
Jean Dubochet.
Dubochet. Okay.
Yeah.
Okay.
So those were the three, Jürgen Frank.
Anyway, Giacomo is here, not too far from where you're sitting.
Yeah.
So tomography is where you tilt the platform. Is that right?
Exactly. So because you know, when all your particles are identical, you just take a picture and you have all orientations. I mean, you can pick up the same. For when they are pleomorphic, I mean, each particle is different. So you need to have different tilts of the
one particle,
of one single particle. But the problem is a dose problem, because if you want to do 20 tilts, you have to divide the dose by 20, the one that you did for the other. So you want to preserve. So, but people are now using only the first few pictures, but you need all of them for alignment.
So do you just do one particle and tilt it, or do you do multiple particles?
Well, on your grid, you will have multiple particles.
One, right?
So you just tilt the grid and you collect and
you will average particles independently. Is that right?
Once you get the tomogram, you identify the subunits that appear the same. If the particle may be bigger, different particles may have different sizes, but you identify the building block, and then you average all of the building blocks and reposition them on where they.
Amazing. It's great. It's great. I heard a talk 2 weeks ago where someone just took protein and did cryo-EM and solved structures of unknown proteins, just blindly.
Yeah.
Instead of producing a protein and solving a structure, they just had an extract of a cell and then averaged and
got it.
And got some structures, and then they could say, okay, what does this look like? What is it? It's incredible. It's like amazing. So do you do mostly cryo-EM now, or is?
No, we're doing both. Of course, we are continuing to crystallize. We also having NMR people and some proteins. The dynamics or changes in conformation are easier to follow by NMR. And we have mass spectrometry, using this, how you call it, deuterium exchange methods to know which regions are protected. So there are many complementing approaches now to the static structures that you get by X-ray crystallography or cryo-EM.
So the field is constantly changing and being improved, right?
Yeah, I think the time dimension is extremely important. So you see, what I was saying is breathing. I mean, we could see the structure of a flavivirus particle, but it's only when you incorporate the fact that it's changing conformation opening and close that you can understand the biology.
Can you see that in a cryo-EM? How do you cannot, but for instance, you have to select the particles that are most closely that form a single class, but you can focus, you can identify different classes and particles in different stages. Use that. And then you can make some assumptions about which is preceding what.
Exactly.
Right?
And now then you can combine that with sort of FRET or some other techniques that will show you changes in distances between two regions and follow the dynamics.
Got it. One last thing I wanted to ask you. So one of your recent papers, 2019, orthobunyloviruses.
Yeah.
You looked at the spike. Did you do just the spike? I guess so, right?
Yeah.
You made the protein, you got crystals, and you looked at this. So was that what made you do that? What was the...
No, but because this was Schmallenberg virus.
Okay.
This virus. So the thing is that we're intrigued by this. They have very big spikes. And the fusion protein, which is in Bunya virus, is called GC, is the equivalent to the E protein. It's twice the size of the flavivirus, one of the others. And actually, Richard Elliott had shown that you can remove the first half of that protein and you could still enter cells. So apparently the fusion protein will be the C-terminal part. And then, but the cryo-EM structures were tomography also at low resolution, and we couldn't tell what was what. So we did the structure of the first half and found that is a head domain that it's making these spikes with the legs. But what was interesting, but we saw that with that same domain, we could deplete all of the neutralizing potential of the serum of animals who were, so all of the antibodies are directed to that, and the fusion protein is not affected, so it doesn't need to change. And there are some orthobunia viruses like lacrosse virus, and there are many cases of fatal encephalitis of people who are immunodepressed by some of these viruses. And now we know that this head domain, which is quite small, could be from people who have antibodies. We could deplete them, have antibodies that will target that and protect people from these infections.
These are class 2?
It's a class 2 protein, but it's a funny one that has an extension that is twice as much, and it does this spike. And that spike it's there in order to distract the immune. I mean, all of the immune system goes to that, so it can be very variable. Whereas the rest of the fusion machinery stays the same.
So the spike is not the fusion domain at all.
No,
right? Fusion domain is
so
elsewhere.
we now have the structure of the second half of the protein, and it looks just like the class 2.
Amazing. Okay. I'm looking at pictures of it here. This little head sticking up.
Exactly.
It's trimeric, right?
It's a trimer, yeah. Huh.
It's very cool. And this was crystallography, right?
That was crystallography, but there was a previous study by Juha Huiskonen, who's doing tomographic reconstructions, and so our X-ray structure fits very nicely into those spikes.
And this is important because it informs vaccine development.
Yeah.
And that
exactly.
In animals, it's a big problem, this virus. So,
and as I was saying, for humans also, this is ways if we had antibodies to treat against these orthobunia viruses for people who are immunosuppressed because it can be very serious. Yeah.
There have been some human infections.
There have been
and
quite a number.
These people are all immunosuppressed who get serious disease.
They get, and in particular, this virus.
Interesting. So you've solved a lot of different structures. Do you have any favorite?
No. What I'm fascinated is by the way of using structural biology. Like it's like doing paleontology without fossils. So you can, and then when the first structure of TBSV and then the cowpea mosaic virus were solved, the surprise was they were almost identical, the same jelly rolls, and now it's found on almost every virus. And now it's the same for this fusion protein. So you can trace back. So that there must have been some very ancient ancestor of these proteins that we're seeing today over and over, and they're still doing their job.
Yeah. I don't know if you've seen this paper of Michael Rossman's where they can find all these viral structural protein motifs like jelly rolls and HK. What is the HK29?
HK97.
97.
Is this phage. Yeah.
They can find it in cellular proteins.
Yeah, in cellular proteins. Well, this is what we are doing in this class 2 proteins. We are now finding in cellular proteins.
Yes.
Do you know what we would like to... We know that they all have the same structure, although there's no sequence similarity in the conservation. Right. So what we would like to do is see if we can use some machine learning approaches now, see what is that they have in common, that then we could just go to the database and identify this is a good candidate for it. So we don't know what is the gamete fusion protein in mammals or in humans. So it has not, so much research on that, but we don't know. And there are many other fusion proteins. We know that exosomes go out of cells and can fuse into other cells, but how are they fusing? What is the protein?
So do you think someday we'll be able to look at a protein amino acid sequence and solve the structure from that?
No, not solve the structure, but you could be able to say, hey, this one it has a very high score to be of this particular class or not. So you probably, at least at the beginning, you will need to go and prove it and solve it and there. But we are beginning to see that this is how the HAP2 protein was scoring quite favorable on some criteria that we have now for the class 2.
Okay. But you think that we
should be do in a systematic way. And maybe, we're looking for a class 2, but maybe it's a class 3 or a class 1, or I don't know. So what we know is there are not too many different kinds of proteins that can do this job. So it's very likely that for other essential functions, we are using them.
Because Michael Rossman and Wayne Hendrickson, who's here, and others have always said, if we have enough protein structures, then we'll be able to look at any sequence and know how it's folding. But you don't think that's going to be?
Yeah, the thing is we continue to find more. So when we have sequences that don't look like anything, we still don't have something that can tell us. And so this is the problem with the metagenomics, all of these viruses, that the problem is that they don't look like anything that we know. So if we knew the structure, we could probably relate them. Yeah.
There are just so many genes whose proteins we have no clue what they're doing.
No clue, yeah.
And because there are so many, it's not likely that we'll ever figure it out.
Yeah.
There aren't enough people working on it. Right? You would need many more scientists than we have. So do you view structural biology as a continuing, growing field?
Oh, yeah. I think we're especially with the developments now on cryo-EM and sort of tomography and the fact that you can do cells, you can study structures in the cell. There are these techniques, FIB milling, that focus ion beam, that you can cut thin slices of a cell and look at the structure inside.
Right.
So I think this, in the next 10 or 15 years, this is going to explode. I mean, everyone is going to be using this method.
So you continue to train people who then go on and have their own labs and do this, right?
Yeah, we're now learning. I mean, cryo-EM was quite new. I was doing X-ray crystallography, but for many of our projects, it's obvious that we will need to move do that. So for 20 years we were trying to crystallize the coronavirus spike protein. Right. It's huge. Right. It has, each subunit is 1,300 amino acids. And then as soon as this cryo-EM came, we in a few months we got that structure to 3 angstroms and everything. And so with David Wessler, that was huge.
So you were at the laboratory for structural virology at the Pasteur, right?
Yeah.
You're not the only structural scientist, structural biologist.
There are others. So but in the virology department, when I went there, I created this unit of structural virology because I'm interested. But you have structural biochemistry. I don't know how they are called. There are these people. There is a department of structural biology. So I'm affiliated. My second affiliation is to that one. But I'm mostly. The problem is that they are not interested in viruses. So I much rather be in the virology department talking to.
Who else is in virology department?
Well, the head of the virology department is Sylvie van der Werf.
Okay, I know her from polio days.
From the polio days, but she's working in influenza
now. Yeah.
So there is Olivier Schwartz, who's working on cells, immune cells. So he was working on HIV, and he has this paper on the IFID-M, these proteins that block fusion. So Simon Wynne Hobson, who HIV, but he's now turned into cancer, and he worked on papilloma and others. So there are like 16 of this, Carla Sale, Marco Vignuzzi, they're in virology. Okay. They are there.
Wonderful.
And there is Michaela Trudwin Muller, who was with Françoise Barré-Sinoussi, and now she's independent, working on HIV. Well, I could continue.
It's good. It's a good department.
Yeah.
Very nice. All right. Let me ask you a couple of fun questions to end. So if you had not become a scientist, what would you have done with your life?
Well, I don't know. I mean, I don't think I could have done something else. So science it really fits the way I am myself. I wouldn't have been good at doing a job, the same thing every day, or it's, I never really thought what I would have become if I had not done science.
Okay. If you could have dinner with anybody, living or dead, who would it be? Anybody.
Dinner with anybody. Wow, you got me off guard here.
It's not something we think about a lot. I
didn't.
Maybe as a physicist, you would pick a physicist, right? Maybe Robert Feynman or Richard Feynman.
Richard Feynman. Oh, that would have been fun, yes.
Wouldn't that be fun? Yeah.
Yes.
Yeah.
Richard Feynman.
RNA or DNA?
my favorite. I say RNA.
Okay. All right. That's a special TWiV on this wonderful Saturday in New York City. You can find our show notes at microbe.tv/TWiV. Send us your questions and comments, twiv@microbe.tv. And if you like what we do, consider supporting us financially. Go to microbe.tv/contribute. My guest today from the Institut Pasteur, Félix Rey. Thank you for joining us,
Thank you.
Today.
It was a pleasure.
I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode was recorded, edited, and posted by Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.